Good evening, all.
Good evening,
Audio
sir.
visual, audio visual clear.
Yes, sir.
Okay. Last day in my class, we are discussing the three acellular organisms, right? Acellular organisms, and these are virus, viroids, and prions. So today we discuss in details the virus because viroids, prions, these are already done. What are the materials they have and how they transmit? Already done. Virus, we are reading details. Viruses. Already some points, some features of viruses are discussed in previous class. With that note, we started virus classification first. Virus classification. Normally, viral structure contains what materials? What are the materials that compose the virus? Genetic materials. It could be RNA, DNA. Okay. Or আর কি আছে? নিউক্লিক অ্যাসিড ছাড়া আর কি থাকবে? Protein. That means two materials compose the virus structure. That's why you are calling nucleoprotein, right? The word is coming nucleoprotein. That means nucleic acid and protein. These two are the ultimate part that make up the virus body. So virus structure, virus classification. The first way to classify the virus based on capsid symmetry. Based on capsid symmetry. We all know that what is capsid. Capsid is the outer layer of the virus that make up the protective layer, and it is composed of protein. And capsid actually a combination of polymerization of protein, and the monomer unit is called capso. What is the word? Capsomer. Capsomer. So based on the capsid symmetry, so symmetry means how does the capsomer are organized to make up the capsid? কিভাবে organize আছে? Okay, so capsid symmetry based on we can classify the virus. Number one, helical symmetry. Number two, icosahedral symmetry. And number three, complex symmetry. Helical symmetry means capsomer are arranged helically to make up the capsid structure. Arranged helical. Icosahedral, it gives a proper definite geometrical shape of icosahedral shape. And the third types, complex. Complex means this type of body structure, capsid structure contain both. That means both type of symmetry is present, helical as well as icosahedral. Helical symmetry, good example, tobacco mosaic virus, TMV. Icosahedral, good example, poliovirus. And complex, good example, bacteriophage. Good example, bacteriophage. in the next section we are describing the bacteriophage structure, then we can easily see that how does the complex symmetry is formed in bacteriophage, because bacteriophage in body structure contain both type of helical and mixed up with icosahedral symmetry. Here, so it you will see in the next section how does it form bacteriophage structure. So type number one, basis on capsid symmetry. Clear, online students? Capsid symmetry based
Yes, sir.
viral classification. Type 2, more meaningful way to classify the viruses. Number 2, basis on nucleic acid. Based on nucleic acid. Yes, it is meaningful because virus contain different types of genetic material in their structure. They contain DNA, they contain RNA. The only existed organism that contain RNA as a genetic material is the viruses. So various type of nucleic acids are present inside of the virus. That is why the more meaningful way to classify the virus is based on nucleic acid. And this is done by actually 1907 David Baltimore. And that is why this classification system is called Baltimore Classification System. System of viruses. This system is well accepted and more meaningful also. Baltimore classify the virus based on the nucleic acid, total 7 classes. Total 7 classes. Class 1, class 2, class 3, 4, 5, 6, and class 7. The class 1 is double strand DNA virus. Class 2, single strand DNA virus. Class 3, double strand RNA virus. Class 4, positive single strand RNA virus. And class 5, negative single strand RNA viruses. Class 6, positive sense but positive sense is a single strand RNA, but they are retrovirus. Positive sense single strand RNA retrovirus. This including class number 6, and class number 7, reverse transcribing DNA virus. Each class is specific for each virus's type, each virus's RNA type. Do not jumble up or skip, because class 1
Sir,
means double strand DNA virus, only double strand DNA virus. You cannot write class 1 means single strand DNA virus. It is the Baltimore classification, and the class 1 is specific for double strand DNA, class 2 specific for single strand DNA. Yes.
sir, what is positive sense single strand?
Have two words are given here: positive sense, negative sense, and other is positive sense retrovirus. We are coming into that. It is basically based on the genome replication strategy. How does the virus replicate? By that way, the name is given. Clear? So we shortly write it here. How does the each of the viruses are replicate? Virus replication of each of the class. Replication means duplication of its genome. Duplication of its genome from the early stage, early classes, that means 11, 12. We know that only DNA is duplicated. No, RNA also be duplicated. In case of RNA virus, the RNA is duplicated also. That means RNA also replicate. We only still know that DNA is replicated. Yes, that is very true. DNA is replicated, and most of the organism DNA is replicated, but inside the RNA virus, RNA also replicated. So various type of replication strategy differ in different classes. That's why the next column is given. How does each of the class is replicated? Simple strategy. Class 1, that is a double strand DNA virus. It mimics the normal mammal's DNA or any kind of organism DNA, double strand DNA. That means it replicates by the double strand DNA convert into another molecule of double strand DNA. It is very simple one. Just like us, our DNA, double strand DNA convert into double strand DNA by means of DNA polymerase, etc. Other enzymes also involved, but the main replicative enzyme is DNA polymerase. Is separate thing. But normal scenario, in case of class 1, double strand DNA replicate into double strand DNA. Correct? But the complicacy here in the class 2. Here it is a single strand DNA original. So how does it replicate? Single strand DNA, first it convert into double strand DNA. Then the DNA is separated to form single strand DNA. The behavior of replication here it show in a direction. First ssDNA convert into double strand DNA. Now double strand DNA is separated and form single strand DNA. That is the strategy of virus replication of class 2. Clear? Because it's originally it is single strand DNA and must convert into single strand DNA after intermediate step double strand DNA. Stage clear? Replication strategy behavior of class 2 clear? Online students.
Yes,
Now come
sir.
to the point double strand RNA class 3. Here originally what present double strand RNA. Double strand RNA first convert into single strand RNA, ssRNA. SS means single strand, DS means double strand, and then ssRNA are conjugate or bind together to form double strand RNA. The final outcome and the initial presence should be same because it is replication ultimately. Replication, the final outcome would be the same molecule. Yes or no? That's called it is replica. রেপ্লিকা মিন্স কি, যদি আমরা জাস্ট কথায় বলি না জেরক্স করে নাও, দ্যাটস কল রেপ্লিকা করা। আমার কপি জেরক্স করছো মানে কি করছো, রেপ্লিকেট করছো। তুমি কি নিজে কিছু করেছো ইনক্লুড? কিছু করোনি, মডিফাই করোনি, আমার কপিটাকে নিয়ে রেপ্লিকেট করছো। সো দ্যাটস ওয়াই দ্য ইনিশিয়াল অ্যান্ড দ্য ফাইনাল উড বি দ্য সেম, and the strategy is different. Double strand RNA first convert into the single strand RNA, then single strand RNA combine to form double strand RNA. Now come to the point positive strand single strand RNA. It is written by plus ssRNA. Plus ssRNA first convert into minus RNA, ssRNA, then plus RNA. Plus to minus, minus to initial product, that is plus. Minus plus minus, correct. Minus single strand RNA first convert into plus RNA, then further convert into minus RNA. But the behavior of class 6 is different. Though it contains class cells RNA, but it do not follow the class 4 strategy. It follow by retrovirus OA strategy, means it retro-transcribe into DNA. See, in case of class 4, there is no DNA present. But in class 6, the RNA, single strand RNA, plus 10 single strand RNA first convert into DNA. RNA double strand DNA. Then it convert ssRNA. So that is the strategy of class 6. They convert DNA structure. That's why they are called retroviruses. Retroviruses have the feature that can convert the RNA into DNA molecule. That is done by the class 6. ssRNA convert into dsDNA and DNA convert into the RNA. At class 4, there is no DNA present. So that's why two classes are different. We have previously mentioned that it is more meaningful and well accepted model of classification of viruses. And every virus is now given nucleic acid structure, either plus, if RNA, single strand RNA, either plus, either minus. So it is well accepted and still it is used. Clear? This one clear, online students? This one clear, class 6?
Yes, sir.
If it is clear, now say, what about the strategy of class 7? It is reverse transcribing, that means it follow the pattern of retrovirus property, but it is originally DNA virus. DNA থেকে হয়ে আবার DNA তে যাবে, মাঝখানে RNA হবে। মানে কেসটা দেখো, dsDNA দিয়ে স্টার্ট, it started with the dsDNA as a genetic material, then it convert into single strand RNA, and RNA reverse transcribe into double strand DNA. So unique property, unique feature. Normally, the class 1, which contain double strand DNA, it directly from double strand DNA without any intermediate of RNA. Check that. But class 7, they are different. They are different in that they have reverse transcribing property. Means double strand DNA convert into single strand RNA. It's an intermediate product. After that, ssRNA convert into the initial double strand DNA. They cannot convert in directly double strand DNA into double strand DNA. Is it clear to all this replication property pattern of all classes?
Yes, sir.
And this question is important. Give the Baltimore classification of viruses. This question is important. It is 3 or 4 marks question. Example of each class. Double strand DNA virus. Example, adenovirus. Class 2 virus, example parvovirus. Double strand RNA virus, example reovirus. Positive single strand RNA, coronavirus. Did you hear the term before coronavirus? Coronavirus is the actual RNA is positive sense and it is RNA virus. Positive sense RNA virus is the coronavirus. Class 5 orthomyxovirus. Positive influenza হয় class 6. Class 6 example HIV retrovirus which contains single strand RNA. Class 7 example hepatitis virus.
স্যার 5 এরটা কি লিখেছেন?
5 হচ্ছে অর্থোমিক্সোভাইরাস ORTHOMYXO অর্থোমিক্সোভাইরাস. So based on nucleic acid structure classification with example done, which is called Baltimore system of classification. Any question? Can we move into the third system, third classification? Any question? All clear?
Yes, sir.
Okay.
Yes, sir.
Third type. Based on envelope present. Based on envelope present. Present or not? So first of all, we need to know the what is envelope. What is the viral envelope? What is viral envelope? Normally, virus structure composed of protein means capsid and nucleic acid. But some virus contain outside a lipid layer. A lipid layer, outer layer, that's called envelope. So what is envelope? Envelope is the outer lipid region present in some virus. And when envelope is present, these viruses contain some spike protein inside the envelope. So spikes protein present or embedded in envelope region. So envelope is the outer portion, lipid portion, and from inside the lipid portion, there is some embedded spike portion or protein. And spike protein can be of two types. One is called hemagglutinin, or shortly it's called HA, or neuraminidase, shortly called N. There are variety of hemagglutinin and neuraminidase. It can be numbering, it given in number. The types of hemagglutinin and neuraminidase given by numbering. HA1, N1, HA2, N2. That means it's numbering or types, subvariants of hemagglutinin and neuraminidase. ছিল দুটোই ছিল number is different. Number is different. Like pandemic species 1, H1N5, like H1N5, swine flu. So number is given based on the subtype, subvariant of hemagglutinin and neuraminidase. So when the spike protein is present, when the when the spike proteins are present because of the envelope structure. When envelope is there, spike protein also be present. And two types of spike protein, hemagglutinin, HA, and neuraminidase. So classification based on envelope, those contain envelope are called envelope virus, and those do not called naked or non-envelope virus. So two types. Envelope virus and type 2 naked or non-envelope virus. Normally, non-envelope virus, if it is icosahedral symmetry, and inside there is a genetic material, either DNA or RNA, that is a normal scenario of non-envelope virus because no envelope structure is present. But when this virus contain envelope, it looks like that. Nucleic acid, and outside of the capsid there is another layer. There is another layer. Here it is the green color layer, that is lipid rich region, and this lipid rich region is called envelope, that cover the whole virus. And inside the envelope there are some protein also embedded. Capsid, this one capsid symmetry তো icosahedral এখানে, এই যে এটা ক্যাপসিড, ভেতরে নিউক্লিক অ্যাসিড হচ্ছে রেড, black capsid, red nucleic acid, envelope, and these are some spike protein. Two type of structure, one is hemagglutinin, other is neuraminidase.
The biota spike protein.
Spike protein, red one, spike protein, two type of conformation is given, one is hemagglutinin, other is neuraminidase. So in case number 2 there is no spike protein, there is no lipid layer, that's why they are called naked or non-envelope. Coronavirus is which one category? Did you know? Huh? Envelope virus. Envelope virus, coronavirus. Influenza virus. Naked virus, example, poliovirus, adenovirus, etc. Classification number 3 based on envelope. Clear or not?
Yes.
Understood? Okay. So virus classification done. So three types of classification more meaningful way is the nucleic acid basis of classification. And next topic, virus life cycle. Virus life cycle. Common life cycle strategy of virus. Viruses are different types, but all gross or common types of common types of life cycle strategy are four events, contain four events. Event number one, attachment. Event number two, entry. Event number three, synthesis. And event number four, exit. Four events of common viruses strategy of life cycle. AESE, more simple way. A means attachment, E means entry, second E. Third, S means synthesis, and last E means exit. So first attached with particular host, specific host. First virus attached with particular host. So attachment is very key things of a disease establishment or infection process. Because without attachment, step number 2, 3, 4 is hampered, obviously. So step 1, that is the specific host attachment, is very important for any infection life cycle of viruses. So attachment here, virus first attached with specific host cell. Now you say, you argue with the search. There are many cells in our body, but virus only affect particular host. How did it achieve? And it is achieved by this specific interaction achieved by receptor ligand binding. Specific attachment is achieved by receptor ligand binding. So virus first attached, and this attachment is very critical. And this for this attachment it needs receptor ligand binding. That means various types of cells are there, but only certain types of cell contain certain receptor. Yes or no? The receptors will vary. Many cells may be present in a host, but the receptors of particular cell is vary from other. Yes or no? So this receptor is recognized by the virus. So virus contain the ligand and the receptor is present by the our own or host cell. So receptor present in host cell and ligand is present in virus. So this attachment give the permission of stage 2, that means entry process. If this attachment is failed, the entry will not happen. Clear? Host cell provide the receptor for particular cell and virus contain the ligand. This receptor ligand binding means virus specifically bind with host cell. So there are numerous host cell, but how they particularly choose the particular host cell? By that receptor ligand binding. Clear? Next step, entry.
Sir, one
Uh,
cell may contain that receptor and other cell may not.
may not or different type. May not or
Different type.
different type. Yes, different type. And that is recognized by the viruses.
So for a
So
particular virus, there is a particular receptor.
particular ligand for that particular receptor. Virus system contain ligand and host cell provided the receptor.
Okay, sir.
Clear? So virus contain particular ligand for target receptor present in host cell. And that binding is important for the attachment process. And after that attachment, the event number 2 is started. That means entry process. Attachment is done, then entry is started. The entry needs some enzymatic help degradation because outside, if it is bacteria, outside contain cell wall, cell membrane, so it should be digested cell wall, then cell membrane, then it gain entry of the into the host body. So entry process is done by enzymatic way. Sometime endocytosis process. What is endocytosis? Endocytosis is the phenomena where outside molecule can enter into the cell. And that's called endocytosis. Cytosis means cell, endo means inside. Outside to inside movement of the molecules is called endocytosis. And now the molecule here is virus. Here the molecule is during the endocytosis period is the virus. So cell unknowingly enter the virus inside them by the help of endocytosis. Normally, nutrient molecules are entered, but in that scenario, it is virus is entering. After the enter into the host cell, totally host cell mechanism is captured. Clear? And start to synthesis its own DNA, own RNA, and own protein, and stop the synthesis of host. তো হোস্টের সেলে যা যা তৈরি হচ্ছে সব 12টা বাজে গেল এখন ও তৈরি করবে কি নিজেরটা কার মেকানিজম ইউজ হচ্ছে হোস্টের মেকানিজম so by utilizing host mechanism it synthesizes own DNA RNA protein not the host DNA RNA protein that's the capturing process and this process is called synthesis event number 3 synthesis so here DNA replication transcription of DNA and protein synthesis occur. Which type of protein synthesis is it host protein or that virus own protein? Virus own
Virus.
protein. If the virus own protein is synthesized, these proteins means capsomer, they are organized inside the host cell and make proper virion structure, proper virus structure. Clear? So after replication, transcription and protein synthesis inside the host cell, mature virus particle produce, because all are assembled into mature virus particle. After mature virus particles enough amount production, they are start to lyse the cell and release from this host cell and gain entry of the another host cell. So that's called the event number 4, exit. After enough amount of production,
নিজে থেকে তো অটোমেটিক তো মানে এক্সিট করে না।
হ্যাঁ ওই করায়, ওই সেলটাকে লাইজ করায়, বাস্ট করে, দেন এক্সিট করে, এক্সিট করে আবার নিউ হোস্ট সেলকে গিয়ে অ্যাটাচ করে। But that time the virus will be multiplied in 1,000 or 10,000. Just simple say, if one virus is enter into the bacteriophage, normal bacterial cell, 1,000 of viruses can be produced. So this 1,000 of virus now enter into the 1,000 of new bacteria. So it is multiplied by GP series, not arithmetic. So continue degrade হতে থাকবে। Like এটা ব্যাকটেরিয়া বললাম, in every scenario means every host cell there is the same pattern. After synthesis there is the exit from this host cell, because this host cell is gone, because host cell এর না প্রোটিন তৈরি হচ্ছে, নিজের হোস্ট সেলের তো কোনো প্রোটিন সব বন্ধ করে দিয়েছে, নিজেরটা তৈরি করেছে। So this host cell is died and now enter into the another fresh host cell and capture them. So that's their strategy. So exit after enough amount of production, they lyse the cell and gain the entry of new host cell. Entry into and start the process of events same way. Cycle begin. So these are called attachment, entry, synthesis, exit, events of virus life cycle.
Yes.
AESC. Clear? Virus life
একটা
cycle.
ডাউট ছিল
বলো।
স্যার, বলছি সিনথেসিসের রেপ্লিকেশন, ট্রান্সক্রিপশন আর প্রোটিন সিনথেসিসটা কি ভাইরাসের হচ্ছে?
ভাইরাসের হচ্ছে by utilizing the host mechanism, because virus do not have any mechanism to produce protein, transcribe or replicate. It uses the host system to make their own protein and stop the synthesis of host.
ওকে স্যার।
That's why they are dangerous না, ও ঢুকছে হোস্টের সমস্ত মেকানিজম বন্ধ করে নিজেরটা চালু করে দিচ্ছে,
তারপরে পুরো নষ্ট করে দিই, তারপর
বেরিয়ে আসছে আবার নিউ হোস্টকে এফেক্ট করছে। Clear? So virus, this life cycle is very general way. Now we look the special virus life cycle, that is bacteriophage. More or less same, but there are two types of life cycle in bacteriophage. One is lytic, another is lysogenic. So how they different? We now recognize that. So bacteriophage. My next topic, bacteriophage. Its structure and life cycle. Bacteriophage means bacteria attacking viruses. Bacteria attacking viruses are called bacteriophage. Clearly, their host cell is a bacteria. Bacteriophage structure. First, they have two parts in their structure, head and tail. Head and tail. The head is elongated structure. Icosahedral shape, symmetry. And the head contain nucleic acid. DNA, double strand DNA. So they are double strand DNA viruses. Clear? And the tail portion is highly complex portion, tail portion. They composed of collar region, they composed of central hollow tubes with protein sheath and they composed of base plate. So highly organized and complex structure is the tail portion, contain three parts: collar, central hollow tubes with protein sheath, and base plate. The base plate or the tail, second point, tail contain two type of filament. One is called STF, other is called LTF. Short terminal filament and long terminal filament. Short and long. STF, LTF. So these are the structural overview of bacteriophage. Now look at the structure.
স্যার এসটিএফ এর ফুল ফর্ম কি বললেন?
Short terminal filament, short type filament, sorry. Type filament, long type filament. Terminal not type. So head capsid symmetry is icosahedral, but the tail symmetry, protein sheath, also contain tail protein sheath, and protein sheath is looks like what types? Helical, na? So helical and icosahedral both are mixed together to form the bacteriophage structure. That's why it is classified under capsisymmetric complex. Complex capsisymmetry. So this is head part and tail part.
Sir, could you repeat what you said just now?
Yes, head is icosahedral shape. Yes or no?
Yes.
Yes, sir.
Tail contain protein sheath, central hollow tube with protein sheath, central hollow tube, and spiral or helically arranged protein sheath. Yes or no?
Yes.
Protein sheath is helical, but the head capsid symmetry is icosahedral. So both type mixed type is present. Symmetry, that's why it is under classification number 3, complex type. See previously, it is under bacteriophage is a complex type of under capsid symmetry because it contains both helical type as well as icosahedral type protein arrangement. This is nucleic acid, genomic material, DNA. Outside is the capsid. No envelope is present, that means non-envelope. These are LTF, long type filament. These are LTF. And these are short type,
CF.
STF. And this is held by a structure known as base plate. Total tail structure and head structure is connected by another structure called collar region.
ডিসপ্লের নামের মধ্যে কাজ মানে বেস বা
Base plate make up the filaments, hold the filaments, STF and LPF. During infection cycle, the tail part is involved in the attachment process. First, attached with the bacterial surface by this tail, LTF and STF. And the protein sheet and the head is the composed of DNA, contain DNA, and protein sheet here act as a injectable means it can contract. Protein sheet is contract and highly powerful contraction occur after the attachment, and DNA is released from the hollow tube and enter into host like an injection. Just inject the DNA during the infection cycle, inject the DNA into the bacterial cell. By that way, tail part is attached with the bacterial surface. This protein sheet is a contractile unit, means it can contract and powerful contraction occur, and DNA is now moved into the from bacteriophage head to the bacterial cell inside. Clear? So infection cycle of bacteriophage. My next topic, life cycle. Two types of life cycle present in the bacteriophage. One is lytic, another is lysogenic. Lytic life cycle, lysogenic. Clear? Common example, lytic bacteriophage. T2, T4, etc. T bacteriophage. T bacteriophage. T can be odd or can be even. Here it is given even number T2, T4. Lysogenic good example lambda bacteriophage. Here to start with lytic life cycle, which is more common in case of bacteriophage, and it is more or less similar to the previous life cycle model of general viruses. See that lytic life cycle.
Entry, sorry, attachment, entry, synthesis, exit.
Exit. Bacteriophage bind to the bacterium. With that point, I will also show the visualization. Means this is bacterial cell, and bacterial cell contain bacterial own DNA, and outside of bacterial cell, phage is getting attached. Bacteriophage is attached, and bacteriophage has its own DNA that is red color. I will show here the red color DNA means bacteriophage DNA. So green color means bacterial own DNA, bacterial DNA, and red color means bacteriophage DNA. So color distinction may easy to understand the process. Okay, so first bacteriophage bind to the bacterial outside bacterium. Next, step 2 started. Phage DNA entered the host cell. Only DNA part is entered, and outside host means outside protein structure is remain there. There is no gain of entry of outside structure. Only red color portion means DNA is entered. Point clear? Diagram clear? Online students.
Yes, sir.
Step 3. After the gain of entry, lytic phase, first they do degradation of host DNA. The host DNA is digested. Clear? The integrity of the host DNA is digested. That means some enzymes are released, carry, they can digest the host DNA. And by utilizing this host DNA, they now synthesize their own DNA. Number 4, new phage DNA forms. The raw materials of the DNA is basically same. That is the nucleotide, DNTPs, deoxyribonucleotides. So they convert DNTPs after degradation of host DNA, and by utilizing these DNTPs, deoxyribonucleotides, they make their own DNA. So now phage DNA is formed. So what will be the color of the phage DNA? It is red. So red color is formed by utilizing green is the raw materials, and when it combined, ultimately the structure is looks like phage, not the host. So phage DNA is given in red color. So red color is formed. So more red color DNA produced. From that DNA, they transcribed and synthesized protein. Phage proteins, number 5. And new phage proteins are synthesized. Phage proteins are synthesized, number 5. Already DNA is produced and also start to produce inside this black color body, bacteriophage body, head, tail, etc. structure. Head tail structure means it will be assembled to form the exact bacteriophage structure. Now it's time to assemble. Number 6. Phage proteins and nucleic acid. Assemble together to form mature bacteriophage. That means in point 6, event 6, more amount of bacteriophage are produced inside the host infected cell. All things are assembled together to make up the good amount of bacteriophage containing nucleic acid, proper structure. See that. Proper structure of bacteriophages are produced in the step of 6 after assembly. After considerable amount of production, the host cell will be lysed and released of mature bacteriophage outside to start the cycle again. That's called lytic cycle. So step number 7, lysis and release to start the cycle again. That means this bacterial cell wall integrity lost and bacteriophage now escape. and these bacteriophage start the beginning of new cycle. Every bacteriophage now start to attach with the fresh bacterium, and the cycle of lytic cycle begins. Next round. So this is known as lytic cycle. All points are clear. Any doubt, any question? Online students,
লাস্টের পয়েন্টটা কি লিখেছেন একটু বলবেন?
lysis and release. Mature bacteriophage now release and new cycle is begin. Means after 7 start new cycle of lytic cycle. So diagram words are all are perfectly aligned. ঠিকঠাক আছে, দেখতে পাচ্ছ ডায়াগ্রামের সাথে ইয়ে অ্যালাইন করতে পারছো তো, ডায়াগ্রাম আর যেটা লিখেছি পয়েন্ট। দিস ইজ মাস্ট, যদি অ্যালাইন করতে পারো তার মানে বুঝতে পারছো, আদারওয়াইজ প্রবলেম। So this is the life cycle of lytic means T2, T4, etc. bacteriophage. What about lambda?
Like
Lysogen.
lytic.
Lysogen. So after 1 and 2, first two steps are similar to the lytic. That means T2, T4 that is done step 1 and step 2, which is absolutely similar to the lysogenic also. But step number 3 is different. Step number 3 here, DNA is degraded, but in lysogenic DNA still intact, remain intact, and the bacteriophage DNA is integrated into the host DNA. Check. Let's check. It is diverged and form lysogenic cycle. So lysogenic point number 3. Start here. Prophage forms. Prophage forms. After DNA enter, prophage forms. What is prophage? Prophage mean when phage DNA integrate into bacterial chromosome. That means, let's look the structure now. Bacterial DNA is totally intact, but the bacteriophage red color DNA is now integrated into the host chromosome, host DNA. So from that point, the third point is actually differed from lytic and lysogenic. The third point here, the DNA is digested, new phage particles are produced, but here in the case of lysogenic, no DNA is degraded, only the bacteriophage DNA is integrated into the host DNA. Difference clear, lytic, lysogenic? Everybody. These two, difference clear?
হ্যাঁ স্যার।
Okay. Next point. In lysogeny, point 3, point 4. Chromosome with integrated prophage replicates through cell division. So after entry, when the cell is divided, there also division of bacteriophage DNA, and bacteriophage DNA maintain their own population like that. When this cells is divided by binary fission, DNA also replicate before binary fission, and when DNA is replicate, bacteriophage DNA also be present in new cell. Check that. So before cell division, only single bacterium contain the bacteriophage DNA. After division, two cells will be contain bacteriophage DNA. So their transmission pattern is different. Lytic virus or lytic cycle transmission pattern is different. They degraded the bacteria after producing enough amount of mature bacteriophage, then start the new cycle again. But here it is in different way. It integrates into the bacteriophage when bacteria is divided into the two bacterial cells. These two bacteria also contain bacteriophage by like that. So when these two bacteria further divide into four, all four contain bacteriophage. Yes or no? When these four divide into eight, all eight contain bacteriophage. So like that, bacteriophage will be multiplied, and it is occur silently. It is occur silently. হ্যাঁ না সাইলেন্টলি হচ্ছে না ট্রান্সমিট করে যাচ্ছে না Number 5. In some rare case, like UV induction, when bacteria is treated with UV or bacteria get enough amount of UV damage, that time prophage excise from host and enter lytic cycle. So now check the interesting scenario. When these bacteria get enough damage with the UV, that time the prophage is excised from bacterial DNA. And now this DNA start lytic cycle. This DNA start lytic cycle means step number 3, digest the DNA and make the new form, new phages, and release the phages. Because these bacteria now chance to not survive because it is UV irradiated, means UV damage already done. So now bacteriophage is left from the bacterial cell by like that. So this is the lysogenic cycle.
Sir, then after
Lytic
fifth
system has a
month,
fixed
it
profile
enters the
like
lytic cycle.
the 6 months.
Then genetic material degradation will occur, and then the next steps will continue.
Continue. Degradation of DNA new phase protein, mature protein means lytic cycle, all the points are covered.
মানে 1 2 তারপরে F3 4 তারপরে আবার F3 থেকে আবার করবে।
তার মানে ভাবো কোনো চান্স ছাড়ছে না, যতক্ষণ ব্যাকটেরিয়া ভালো ছিল এটা হচ্ছে সেই ধরনের বন্ধু, যতক্ষণ ব্যাকটেরিয়া ভালো ছিল ততক্ষণ অব্দি ও সাইলেন্টলি ব্যাকটেরিয়ার মধ্যে স্প্রেড করেছে, বাট ব্যাকটেরিয়ার কন্ডিশন খারাপ, লেফট, ছেড়ে দিল, জাস্ট কিক করে বার করে দিল, ওকে ছেড়ে দিয়ে নিউ ব্যাকটেরিয়া মানে লাইটিক সাইকেলে চলে গেল। So lytic cycle, lysogenic cycle of bacteriophage important. Any of the cycle can be given, or question can be mentioned. Bacteriophage life cycle, just bacteriophage life cycle, 5 marks. Both life cycle will be concluded, lytic as well as lysogenic.
তো সেক্ষেত্রে তো আমরা এরকম করতে পারি না, তাহলে আমরা আগে
পুরোটাই করতে হবে।
না না মানে আমরা এরকম বানাতে পারি 1 2 3
হ্যাঁ, কারণ ব্যাকটেরিওফেজ আর লাইফ সাইকেল বলেছে, তো টোটালটাই করতে হবে তোমার।
না না বলতে যে একইভাবে মানে ধরে নিই যেরকম এখানে আছে এটার 1 2 তো সেম, তারপরে এর 3 4 5 আবার এর 3 থেকে শুরু হবে।
ঠিক আছে, তুমি এখানে অ্যারো দিয়ে দিলে মানে এটা এখান থেকে স্টার্ট হবে, ফার্স্ট দুটো স্টেপ তো নরমালি হবে না, ওটা বোঝা যাবে যে ওটা আফটার 3 থেকে স্টার্ট হবে, and this arrow is after 5 means start with 3 4 5 6 7. Clear? So virus overall discussion done. Previous day we discussed about the basic points, some features. Today this point is elaborated: classification, bacteriophage structure, bacteriophage life cycle. So virus structure is ended. So next day we started fungi, cellular organism, fungi.
সিলিন্ডার ওটা তো আমরা না
হ্যাঁ, ওটা ডিফারেন্ট। এত অবধি একটা এক্সাম হবে,
ওটাই তাই বলছি।
হ্যাঁ, এত অবধি একটা নোটস পাবে, মানে ফ্রম ডোমেন সিস্টেম, কিংডম সিস্টেম টু ভাইরাস অবধি একটা এক্সামের টপিক, ওটার একটা নোটস পাবে কম্প্যাক্ট অ্যান্ড একটা এক্সাম, অ্যান্ড নেক্সট ক্লাস উইল বি ফাঙ্গি। হ্যাঁ
স্যার, ডোমেন সিস্টেমের যেটা সেকেন্ড ক্লাস ছিল স্যার, ওটা আমি করতে পারিনি, কেন কি আমি ট্রেনে ছিলাম, আউট অফ স্টেশন ছিলাম। তো ওটার ভিডিও কি একটু তাড়াতাড়ি
দেওয়া আছে,
আপলোড হলে?
ভিডিও আপলোড হয়ে গেছে, দেখো ডোমেনের এই ক্লাসগুলো কোর পেপারের সব ভিডিও আপলোড হয়ে গেছে, এটা হচ্ছে লাস্ট যেটা আপলোড করতে হবে, 2 days ক্লাস
ওর
দেওয়া
আগেটা
আছে।
সব হয়ে?
হ্যাঁ, সব হয়ে গেছে, সেট পেপারটা
আর স্যার,
অনেকগুলো ল্যাগ আছে।
ছিল ওটারও একটা ক্লাস আমার মিস গেছিল, যেদিন গ্রেন বেসড ফার্মেন্টেশন স্টার্ট হয়েছিল, ইডলি ডোসা ওইসব, সেটা মিস গেছিল স্যার। ওই
ইডলি
ভিডিওটা
ডোসার ক্লাস, হ্যাঁ বলছি, ইডলি ডোসার ক্লাসে যে ভিডিওটা না, ওটা অনেকভাবে কেটে গেছে, মানে ওটা রেকর্ডিং এ প্রবলেম ছিল, বাট কিছুটা রেকর্ড হয়েছে, স্টিল ওটা আমি আপলোড করব, এটার পরেই আপলোড করবো, মানে যেটা লাস্ট করেছি সেক পেপারে ক্লাসে তারপরে আপলোড করব, কারণ ওটার টাইমটা অনেক কম হয়েছে, মানে কি কেন কে জানে, যাই হোক ভিডিওটার রেকর্ডিং এ প্রবলেম আছে, তো ওটার মধ্যে জাস্ট ওই ডোসা আর ইডলি ম্যানুফ্যাকচারিং প্রসেসটা পাবে, এর আগের পার্টটা পাবে না, যে গ্রেন ন্যাচারালি কিভাবে ফার্মেন্টেশনে হেল্প করে, হোয়াট আর দ্য মাইক্রোবস ইনভলভ ইন দ্য গ্রেন। এর জন্য ক্লাসে যারা অ্যাটেন্ড করেছিল তাদের থেকে কাউর থেকে একটা গ্রুপে মেসেজ করে নিয়ে নাও, যারা ক্লাস করেছে ক্লাস নোটসটা, ঠিক আছে, ওটা বেটার হবে, কারণ আমার যে ভিডিওটা আসবে সেটা কেটে আসবে, ওটা 15-20 মিনিটসের জাস্ট ভিডিওটা রেকর্ড হয়েছে, ঠিক আছে।
আর স্যার, এটার এক্সাম কবে হবে?
এটার এক্সাম আমি নোটসটা দিই তারপরে, তো জানো তো, মানে প্রসেস তো জেনে গেছো, নোটস দেবো তারপরে উইকে এক্সাম হবে, আপাতত ক্লাস স্টাডি করো, ক্লাসে যা নোটস দিয়েছি বা ক্লাসে যা পড়িয়েছি বুঝিয়েছি সেটা স্টাডি করো, নোটস পাবে, ওটা রিড করবে, 1 উইক পরে এক্সাম হবে, ফ্রম এত অব্দি টপিক টিল লাইসোজেনিক লাইটিক সাইকেল টপিক, ঠিক আছে। এটাও একটা অনেকটা বড় টপিক ছিল, সেলুলার অর্গানিজম আছে, ডোমেন আছে, কিংডম আছে, অ্যান্ড ভাইরাস লাইফ সাইকেল। ওকে,
এর মাঝখানে মানে ওই দেখতে পারে তো 4 নম্বর ডেটটা হয়ে যাবে।
বলছি প্রায়ন প্রোটিন আর নর্মাল প্রোটিনের ইন্টারঅ্যাকশনের মেকানিজমটাকে এক্সপ্লেন করে দেবেন?
3 নম্বর দেখতে পারে 3 নম্বর হয়েছে না 4 নম্বর?
3 নম্বর,
ওটাও তো অনেকটা
ওটা বাকি আছে, এখনো এক্সাম দেবো, নোটস দেবো,
না
বাকি আছে, বাকি আছে,
আপনি বলছেন ওটা এখন।
হ্যাঁ, যেটা বলছো বলো, এনপিআর পিপি,
হ্যাঁ স্যার।
নর্মাল প্রোটিন আর প্রায়ন প্রোটিন, হ্যাঁ, নর্মাল প্রোটিন আর প্রাইম প্রোটিনের মধ্যে ডিফারেন্স হচ্ছে এটাই, ফোল্ডিং এর ডিফারেন্স। প্রাইম প্রোটিনটা হচ্ছে অ্যাবনর্মালি ফোল্ডেড। দেখো, নর্মাল একটা প্রোটিন স্ট্রাকচার খুব ভালো কম্প্যাক্ট শেপে আসতে গেলে ফোল্ডিং দরকার হয়। গুড ফোল্ডিং মেক্স দ্য প্রোটিন গুড স্ট্রাকচার ওয়েল ব্যালেন্স ফাংশন, ঠিক আছে। এবার নর্মাল প্রোটিন আর প্রাইম প্রোটিন মধ্যে ডিফারেন্স কি, স্ট্রাকচারটা চেঞ্জ হয়ে যায়। এবার এই প্রোটিনটা তো স্ট্রাকচার অলরেডি চেঞ্জ, এ যখন এনপির সাথে গিয়ে বাইন্ড করবে, এনপির নর্মাল স্ট্রাকচারকে ও ইনডিউস করবে অ্যাবনর্মাল হতে। মানে ওই যে বলে না, বস্তায় একটা পচা আলু থাকলে আর একটাকে পচাবে, লাইক দ্যাট। যদি ইফ পিপি ইজ প্রডিউস ইনসাইড দ্য বডি, দিস পিপি ইনডিউস কনফরমেশনাল চেঞ্জ। কনফরমেশনাল চেঞ্জ মানে হচ্ছে স্ট্রাকচার ফোল্ডিং এ চেঞ্জ করাবে, আর এনপি কে ও পিপি তে কনভার্ট করে দেবে। এবার এই পিপি অনেক অ্যামাউন্টে যদি তৈরি হয়, এগুলো ক্লাম্পিং হয়, অলিগোমারাইজ হয়। অলিগোমারাইজ মানে হচ্ছে একটার সাথে একটা বাইন্ড করে। বাই নর্মাল প্রোটিন কিন্তু নর্মাল বাইন্ড করে না, নর্মাল প্রোটিন ইন্ডিভিজুয়ালি ওয়ার্ক করে, বাট প্রায়ন প্রোটিন গুলো ক্লাম্পিং হয়ে যায়। এবার ক্লাম্প হয়ে গিয়ে তোমার সিস্টেমের মধ্যে প্রবলেম বা ডিজিজ ক্রিয়েট করছে। আর মেনলি এটা নিউরোলজিক্যাল সিস্টেমের ক্লাম্পিংটা হয়, অ্যান্ড নিউরোলজিক্যাল ডিজিজ গুলো করায়। এর জন্য আমরা দেখো যা যা ডিজিজ পেয়েছি, ম্যাটকাউ ডিজিজ, যেটা হিউম্যানের ক্রুজেপেল জেকব সিনড্রোম, সবই হচ্ছে ওই নিউরোলজিক্যাল টাইপের, মানে এটা অ্যানিমেল বেসড ডিজিজ প্রায় মেনলি।
ঠিক আছে স্যার। আর স্যার বলছিলাম যে আজকে ক্লাসের স্টার্টিং এ আমি কমপ্লেক্সটা মিস করে গেছিলাম, কমপ্লেক্স ক্যাপসিড সিমেট্রিটা, ওটা আবার বলে দেবেন?
ওটা বলবো, তিন রকম ছিল, হেলিকাল, আইকোসাহেড্রাল আর কমপ্লেক্স। হেলিকাল তো দেখতে পাচ্ছ, হেলিকালি অ্যারেঞ্জ ক্যাপসোমিয়ার। আইকোসাহেড্রাল ওই একটা পার্টিকুলার শেপ ছিল। যখন বোথ কম্বাইন থাকবে একটা স্ট্রাকচার, মানে হেলিকাল সিমেট্রিও থাকবে অ্যাজ ওয়েল অ্যাজ আইকোসাহেড্রালও থাকবে, তখন আমরা স্ট্রাকচারটাকে বলবো যে কমপ্লেক্স। যেমন ব্যাকটেরিওফেজে, ব্যাকটেরিওফেজে হেডে ছিল হচ্ছে আইকোসাহেড্রাল টাইপ সিমেট্রি, আর যদি তুমি ওই প্রোটিন শীটটা দেখো যেটা টেলের কাছে ছিল, সেটা হচ্ছে হেলিকাল টাইপের ছিল, যেটা হচ্ছে কন্ট্রাকশন করাচ্ছে। তো হেলিকাল আইকোসাহেড্রাল বোথ মিক্সড আপ ইন আ সিঙ্গেল স্ট্রাকচার মিনস এটা কমপ্লেক্স টাইপ।
আর স্যার বলছি আইকোসাহেড্রাল একটা এক্সাম্পল বলেছিলেন, ওটা মিস করেছিলাম।
অ্যাডিনোভাইরাস বা পোলিওভাইরাস আইকোসাহেড্রালের শেপটা এরকম হয়, এই যে যেটা আঁকছিলাম, মাল্টিপল তোমার ট্রায়াঙ্গেল মিলে স্ট্রাকচারটা তৈরি হয়। এই যে এই ধরনের স্ট্রাকচার, এটা হচ্ছে একটা আইকোসাহেড্রাল শেপ।
মানে আইকোসাহেড্রাল ফর্মে অ্যারেঞ্জ করে, তাই তো?
হ্যাঁ, ওই তোমার ক্যাপসুমিয়ার যখন অ্যারেঞ্জ হবে, তখন এই ধরনের অ্যারেঞ্জমেন্ট নেবে, তখন আইকোসাহেড্রাল বলে।
ঠিক আছে স্যার।
ওকে।